Testing consciousness in brain organoid robots. Ladies and gentlemen, esteemed colleagues and students. Today, we embark on a fascinating journey at the intersection of neuroscience, robotics, and artificial intelligence. Our goal is to explore an experiment designed to test whether a brain organoid robot could exhibit signs of consciousness through goal-directed behavior, specifically seeking water and nutrients. Objective. The primary objective of this experiment is to determine if integrating a human brain organoid into a robotic system can exhibit behaviors that suggest a rudimentary form of consciousness. We aim to observe if such a system can perform goal-directed actions, namely locating and acquiring water and nutrients in a controlled environment. Method. Our methodology consists of several key steps. 1. Setup and integration. First, we integrate a human brain organoid with a robotic platform. This robot is equipped with basic movement capabilities and sensors for environmental interaction. The brain organoid serves as the control center, potentially guiding the robot's actions based on neural activity. 2. Environment creation. Next, we create an environment tailored for this experiment. This environment contains strategically placed sources of water and nutrients. Additionally, we introduce obstacles to challenge the robot's navigation skills, ensuring that any goal-directed behavior is not purely reflexive, but involves problem-solving. 3. Baseline behavior. To establish a baseline, we record the robot's behavior without the brain organoid. This helps us understand the robotic platform's mechanical responses and sets a comparative standard for subsequent trials. 4. Training and conditioning. We then proceed to train the organoid robot system. Using reinforcement learning techniques, the system learns to recognize and seek out water and nutrient sources. Positive reinforcement helps in conditioning the organoid to associate specific neural patterns with successful goal completion. 5. Behavioral testing. In this phase, we conduct multiple trials, where the organoid robot system navigates the environment to locate water and nutrients. We meticulously monitor and record its behaviors, focusing on decision-making processes, problem-solving abilities, and adaptability to environmental changes. 6. Control group. To ensure the validity of our findings, we use a non-biological AI-controlled robot as a control. This control group helps us differentiate behaviors arising from the brain organoid's neural activity from those that could be achieved by conventional AI systems. Data collection. Data collection is a critical aspect of our experiment. We track several parameters. Success rate, the frequency with which the organoid robot successfully finds water and nutrients. Time taken, the duration required to reach these targets. Complexity of problem-solving strategies. The robot's ability to navigate obstacles and adapt to changes. Additionally, we record neural activity within the brain organoid during tasks. Identifying patterns correlating with decision-making processes provides insights into the organoid's role in driving goal-directed behavior. Analysis. In our analysis, we compare the organoid robot's behavior and neural activity to that of the control group. We assess whether the behaviors exhibited by the organoid robot system demonstrate intentionality and adaptation, suggesting a rudimentary form of consciousness. If the organoid robot shows a higher degree of problem-solving ability and adaptability compared to the control, it may indicate that the brain organoid contributes to conscious-like behavior. Ethical considerations. Given the nature of our experiment, ethical considerations are paramount. We must adhere to strict ethical guidelines concerning the use of human brain organoids. The potential for developing sentience in these organoids necessitates careful monitoring and ethical oversight. Collaboration with ethicists ensures that our research remains within the bounds of ethical propriety, while advancing our understanding of consciousness. Conclusion. This experiment represents a significant step toward understanding the potential for consciousness in brain organoid robots. By meticulously observing and analyzing goal-directed behaviors, we aim to uncover whether integrating a human brain organoid into a robotic system can produce signs of a rudimentary form of consciousness. The insights gained from this research could have profound implications for the future of neuroscience, artificial intelligence, and our understanding of what it means to be conscious. Thank you for your attention. I look forward to your questions and a stimulating discussion on this groundbreaking topic.